Hi, everybody. My name is Ed Davis. I'm Senior Application Scientist at GeneCopoeia Incorporated, located in Rockville, Maryland, just outside of Washington, D.C. I'd like to thank you all for joining me for today's webinar entitled Genome Editing, How Do I Use CRISPR? We really appreciate it. Before I go into the webinar, I'd like to go over a few practical matters. First, you may ask me questions at any time. But in order to do so, your microphones will be muted, so you won't be able to speak to me vocally. Instead, you need to type your questions in the questions box in your control panel. If you can't see your control panel, then it might be hidden, in which case look for a small strip of buttons on one side of the screen. One of those buttons will be orange with a white arrow pointing in one direction. If you click on that button, you'll then be able to see the control panel and your questions box. Secondly, in the interest of keeping the talk going smoothly, I will wait until the end of the talk to answer your questions, and I will answer each question in the order in which it was received. When I do so, I will read each question out loud and answer each question out loud for everyone to hear. And finally, I'm going to be sending you a few links for downloading technical notes or for future seminars. And I'll be doing that through the chat box, which is also located in your control panel. So look out for those prompts. Okay, so with that, I'm going to start with the webinar. All right, so here are the goals of this presentation. Very straightforward. First, I'm going to show you steps to take after receiving genome editing plasmids, CRISPR plasmids from GeneCopoeia. And I will say up front that this talk is going to be highly focused on using CRISPR in stable cell lines, but it's possible that you will also be able to apply some of it to other model systems. And secondly, we're going to have a Q&A session at the end. So I'll speak for a little while, and then I'd like to have a Q&A session to make this more of a workshop type of event rather than just a straight-up seminar. So I'd like to start off by telling you who we are at GeneCopoeia and what we're about. We're a company that's focused primarily on functional genomics and cellular biology. And one of our chief product areas involves clones. These are plasmid clones. And they are essentially ready-to-use clones for various applications. We started out as a provider of a large collection of ORF or orphan reading frame clones for humans, mouse, rats, and other organisms. But since then, we've added clone types such as promoter-reporter clones for mammalian promoters, microRNA clones of various types like 3' UTR target, precursor, and inhibitor clones for studying microRNA biology, CRISPR clones, you're going to hear a little bit about that today, and shRNA clones for various knockdown applications. And I will say again that these clones are primarily ready-to-use expression clones, that are oriented usually toward mammalian systems. So you receive the clones from us, and you can use them right away without any subcloning. In addition, we cover viral systems such as lentivirus. We have a lot of products and services for lentiviruses, from clones to custom particles to packaging kits. And we've just recently added adeno-associated virus or AAV type of products. In addition, we have a large number of kits and reagents of various types, such as for transfection. We have a line of transfection reagents. We have luciferase kits for both analyzing gene expression or other applications involving a promoter. These are primarily based on either the Firefly/Manila system or the secreted Ghostia luciferase system. We have a number of FISH probes for fluorescent in situ hybridization. We have indel detection kits. You'll hear about that today. Those are primarily oriented toward genome editing. And we have cloning kits. And finally, we have a number of products for fluorescent detection, such as cell function assays. We have kits for detecting various stages of apoptosis. We have nucleic acid detection kits, cell structure probes. So let's say you want to visualize organelles in your cells. And we also have a large number of fluorescent dyes for conjugating to various agents like secondary antibodies. But of course, you're here to learn about CRISPR, so let's dive into that. All right, so again, I mentioned earlier that a lot of our CRISPR products are oriented toward work in cell lines, mammalian cell lines. And this workflow diagram illustrates exactly what I mean by that. So we have a number of products and services for various stages in the process of modifying genes in cell lines. In the beginning, you'll want to do a number of steps such as sequence your target region. I'll go over this, and design and construct single guide RNAs and if necessary, HDR donors. And for that, GeneCopoeia has large collections of sgRNA and HDR donor clones. Step 2, we talk about also preliminary work for validating single guide RNAs. We use our indel check system. I'll talk about that. And also for determination of chromosome copy number, you'll hear about the VividFish, our line of VividFish fish probes. Then the next stage of the process is transfection or transduction. Well, we have reagents and products for that, such as our transfection or lentiviral packaging systems. And finally, I'll talk about clonal isolation and screening. And again, that'll be using a product line known as the indel check system. So here's an outline of today's seminar. I'm going to first go over genome editing technologies and applications, the cool things that you can do. Next, I'm going to talk about this downstream workflow for how to use CRISPR in stable cell lines. Then I'm going to talk about the importance of functional validation for CRISPR. Then transfection or transduction, followed by clonal isolation. This is a very important concept that I'm going to spend significant time on. Next, I'm going to talk about what to do for screening for edited clones. And finally, things to look out for, various pitfalls that can come up during your genome editing work. All right, so let's talk about the background for genome editing technologies and applications. So one of the main applications for CRISPR, though by no means the only one, is to do gene ablation studies. So for many years, it was always very difficult to, say, get rid of gene functions in higher order cells systems. Like it's always been traditionally easy to do to knock out genes in, say, yeast and bacteria, but to do things in mammalian cells or in even organisms like C. elegans or Drosophila was rather difficult. And then RNA interference came along, which made the process much easier. And a lot of people are familiar with the process of knocking genes down using RNAi. Well, CRISPR can be used for gene ablation experiments as well, and this is very relevant to today's talk, but there are some fundamental differences between the two approaches that are very important to keep in mind when doing your screening work and designing your experiments. So for one thing, both CRISPR and RNAi are designed to change the expression level, of course, in a downward direction in this case. RNAi, of course, does this by a knockdown. Again, you're doing really well if you get, say, 90% knockdown. Often you get less. Sometimes that's enough for for understanding what your gene is doing in a particular situation, but you do have some residual gene product left over, so that can complicate your results. On the other hand, CRISPR for gene ablation studies is designed to knock genes out, completely knock them out. And it does this by changing the genetic code, makes a permanent change in the genetic code, which is not the case with RNAi, where, of course, you transiently or temporarily alter post-transcriptional or post-translational gene expression. But with CRISPR, and again, this is very important, you'll hear about this, you pretty much need to do clonal isolation, at least in cell lines, to get a clean result, which is not the case with RNAi. So again, it's very important to keep this in mind when undergoing your CRISPR work. And again, I want to emphasize the importance of clonal isolation. Now, that was a very brief overview between the differences between CRISPR and RNAi for gene ablation studies. If you want to know more, we have a technical note on the topic shown here. This is the download link, but I don't expect you to remember that link or write it down. Instead, I will copy and paste it into the chat box that I mentioned earlier. And I just sent it to you in the chat box, so look for that. You can click on it at any time you want during the seminar. Okay, so let's get back to the background on CRISPR. So most CRISPR events begin with targeted DNA editing by double-strand break or DSP induction. Okay, so this is not true of all CRISPR experiments, but it's true of most. So most events start by directing CRISPR, and I have TALEN here as well. It's an older technology, to a given site in the genome. And what happens is when this is properly targeted, CRISPR will cut the chromosome, make a double-strand break. Okay, and these breaks must be repaired or cells will die. So there are two major mechanisms for repairing these breaks. The first, shown here on the right, is known as homologous recombination, also known as homology-directed repair. This is essentially what happens during meiosis and DNA damage, where you get crossover between a copy of the broken chromosome and the broken chromosome itself. But also, this can occur when you have an exogenously added donor DNA, shown here. You get recombination between homologous arms flanking the break site. And when you get this, you copy the information from the donor DNA, serves as a template, to the broken DNA. And what you end up with is essentially a knock-in of any sequences you put in between these homologous arms on either side of the break. So this can be used to knock in virtually any site, like a fluorescent marker or a point mutation, for example. On the other hand, in the absence of a homologous template, and this is what happens in usually in non-dividing cells, but also in other situations, you get a process known as non-homologous end joining, or NHEJ. NHEJ is a process whereby you simply stick the broken ends of the chromosome back together. Now, this is significant because NHEJ is a highly error-prone process, especially when compared with homologous recombination, which has a much, much lower occurrence of errors than NHEJ. So what happens with NHEJ is you get small insertions or deletions during the repair process. This will often lead, at least 2/3 of the time in theory, to frameshift mutations, which usually knock genes out. So this is a very useful and simple and quick method for getting gene knockouts. So now the most widely used, though by no means the only CRISPR system in use right now is the CRISPR-Cas9 system from Strep pyogenes, the bacterium Strep pyogenes. Now, shown here is essentially what this system looks like. It's a two-component system, also known as an RNA-guided endonuclease. And the way this works is you have, again, two components. One of these, represented by this yellow shape, is known as the Cas9 nuclease. Okay, and this forms a complex with another entity known as a single guide RNA, or sgRNA. sgRNA is a chimeric RNA that contains target site information, which is shown here in blue. So when you have a target site that you program into this system that occurs upstream of an NGG PAM site in the genome, this complex will find this site in the genome, hybridize with the strand opposite the PAM site, and then create a double-strand break in the chromosome. Now, as I mentioned before, these breaks will be repaired by either non-homologous end joining or homology-directed repair, and it works very efficiently. And it makes a cut just upstream of the PAM between bases 3 and 4. So, okay, so that's a very basic overview of how CRISPR works, at least mechanistically. Now, what about applications? What are the cool things you can do with this system? Well, there are quite a few, actually. These are some of the most common. I mentioned gene knockout twice here because gene knockout can be done either through non-homologous end joining that I mentioned before or using a plasmid donor. You can also do correction of disease mutations Let's say you have a genetic disease that's caused by a point mutation. You can correct that mutation and bring it back to wild type. There's also engineered disease resistance. This refers to studies going on where people knock out the CCR5 gene to cure patients of HIV. There's clinical trials for that technique going on. You can do forward mutagenesis screens using pathway-specific or whole genome single guide RNA libraries. There's in-frame fusion tagging. So, like one published example is to put a C-terminal tag on of a gene with V5, or you could use a fluorescent marker to tag your gene. And then there's also transgene knock-in, that is knocking in transgenes to specific sites known as safe harbor. All right, so now that I've given you that overview, again, this is not a seminar really that's about CRISPR itself, but about how to use it. Now I'm going to talk about this downstream workflow, how to use CRISPR. So here's a figure that shows an overview of this downstream work. It involves several steps. We start off by making bacterial stocks for plasmids, very mundane but important. Then we have steps for doing both functional validation and target copy number determination. I'll go over that. Followed by transfection or transduction if you're using lentiviruses, selection or sorting, isolation of single clones. Again, I want to emphasize single clone isolation a lot. And then the screening phase using PCR or the T7 endonuclease 1 assay and sequencing. All right, so step one, I mentioned make a bacterial stock. Again, this is a very mundane step, but you may receive plasmid DNA from GeneCopoeia, and which is ready to use. But we always recommend that people make a bacterial stock so they have a renewable source of the plasmid. Naturally, we could always make more for you. We'd be happy to do that, but you can put these plasmids into bacteria. And again, this is necessary to make a renewable stock without reordering DNA preps. The plasmids are circular and can transform E. coli using standard methods. They have standard E. coli origins of replication, as well as ampicillin, usually ampicillin resistance gene for selection. So we recommend, again, making a bacterial stock of the plasmids you receive from us. Okay, now I'm going to talk about functional validation. So what do I mean by functional validation? Okay, well, this comes in several different formats. So now, by default, the single guide RNAs are not functionally validated. We use the reference, we design them to using the reference sequence, the NCBI reference sequence. So, and they're not functionally validated. So, the first thing to keep in mind is that there could be polymorphisms in your particular cell line that could disrupt, decrease, or eliminate CRISPR activity. Okay, so we recommend that you sequence the target in your cell line just in case there is some kind of polymorphism in the target sequence. Okay. And secondly, there's the part about functional validation itself. So again, even if the sgRNA target sequence matches 100% with the target sequence in your cell line, there's a lot of variation among different CRISPR sgRNA targets. So now the reason we recommend doing this is because some obviously cell culture-based genome editing projects can take several months, 3 or more months to complete. So of course, if there's a large variation in efficiency between different single guide RNA targets, even with good algorithms that GeneCopoeia uses to design these, we still recommend doing functional validation because you could occasionally get a CRISPR clone that does not work or works very poorly. And that's why I have here, not all CRISPR sgRNAs are created equal. So we recommend testing the function of these first before undergoing these long genome editing projects to avoid wasting time, potentially wasting time and expense. And what we recommend doing is a cell culture-based strategy using the T7 endonuclease 1 assay. So this is a schematic of how the T7 endonuclease 1 assay works. It exploits the fact that non-homologous end joining leads to these indel mutations that I mentioned before. So it's a PCR-based and then assay followed by digestion with an enzyme known as T7 endonuclease 1. So what happens is when you make a PCR, when you do PCR on a transfected population of cells that have been transfected with CRISPR plasmids, you often have a mixed population of mutants. These are indel mutants as well as unmodified chromosomes. And so when you denature and reanneal these, you then get homoduplex DNA that can be either a mutant over mutant or wild type over wild type, or you get heteroduplex DNA, which can be mutant over wild type or different one mutant A over mutant B, because non-homologous end joining is random, so you get different kinds of mutations even at the same target site. And these heteroduplex DNAs are substrates for the T7 endonuclease 1 enzyme. So GeneCopoeia has developed a system, a series of kits known as the indel check T7 endonuclease 1 system, and it exploits this property of CRISPR to create these indel mutations, and then when you do this denaturation and reannealing step, you get heteroduplex DNA. So again, on the left shows the basic overview. You get CRISPR causes a double-strand break, and then if you design primers that flank this break site and do PCR on a bulk population of cells, you can then digest these PCR products with T7 endonuclease 1 in the kit and run them on a gel. So a typical gel is shown here. These two lanes are the PCR products generated from here that are either untreated or treated with T7 endonuclease 1 from the indel check kit. And if everything went well, if your CRISPR single-guide RNA worked properly in the cell lines and all the reagents are working well, you will see the presence of 3 bands of predicted sizes. One of these will be the uncut band. This usually results from homoduplex DNA, which is not cleaved by T7 endonuclease 1, as well as 2 bands, smaller bands that are faster migrating bands, shown here denoted by the asterisks. So if you see this, then you know that you had a successful CRISPR experiment, and you can go forward with confidence that everything is going to work quite well. So the T7 endonuclease 1 indel check system contains all the PCR and T7 endonuclease 1 reagents that you need, and you can even purchase these with or without target-specific primers right from our website. Now, if you don't want to do the CRISPR functional validation yourself, then we can do it for you using GeneCopoeia's functional validation services. This is an additional service available for human and mouse single-guide RNAs. We use the same assay, the T7 endonuclease 1 assay that I just described. We will transfect model cell lines for either human or mouse. In the case of human, it's HEC293, or mouse, we use Neuro2A cells with your custom single-guide RNA plasmids. Again, we do the T7 endonuclease 1 assay system, and we provide you, when it's all done, with a validation report. All right, so if you want to know more about the power of the indel check system, we have a technical note on the topic shown here, and this is the download link. But as I did previously, I will copy and paste this link into the chat box, and you can download the link at your leisure. Okay, so I just sent it to you in the chat box, and if you don't see it, then you may need to scroll down a little bit. Okay, so the next step in the process is copy number determination. So why do copy number determination? What do I mean by that? So for complete knockouts or mutagenesis, you might need to modify all alleles. And you notice I said all alleles and not both alleles. And that's because not all cell lines are diploid. Some lines, like HeLa, for example, have 3, 4, or more copies of the genome. And so now you might expect multi-allele modification to be more difficult to achieve than single allele. However, there's at least one publication that shows that double allele modification actually does occur more frequently than single allele. Nevertheless, you can, and we have seen this, you can get single clones from cell lines that have been treated with CRISPR that have 1, 2, 3, or all possible combinations of gene modifications, gene modifications. So we strongly recommend determining the number of copies that your gene is present in your cell line while you're doing your functional validation. And the best way to do that, we feel, is using fluorescence in situ hybridization, or FISH. And I'm going to go over an application where we used FISH with a CRISPR knockout. And so this is a project that we did for a customer a while back, which was to knock out the histone deacetylase 6, or HDAC6 gene in the human MH7A cell line. HDAC6 is a gene located on the X chromosome in humans, and MH7A cells are human immortalized synovial fibroblasts. So when we took this project on for the customer, we asked them, well, how many copies of your gene are in the cell line? And we got, because there was no information from the literature or from ATCC about it, and so the customer actually did not know. So what we ended up doing was we used a GeneCopoeia FISH probe that targets the centromere of the X chromosome to get an estimate of the copy number of this gene in this cell line. So this figure shows the results of that test. The top panels are cells that are from the MH7A cell line for the customer, and the bottom set of panels is from the cell line HT1080, which is a known human diploid cell line. And when we probed both cell lines with our vivid FISH X chromosome probe, this is what we saw. We found that most of the MH7A cells that we examined had five signals, or 5 green dots, whereas the HT1080 cell line only had 1. And it didn't have 2. The HT1080 cells don't have 2 because it's a male cell line, as we see from hybridization with a Y chromosome probe. But the MH7A cell line does not hybridize with the Y chromosome probe, so it's a female line. But the most important result here is the fact that we discovered that this cell line has 5 copies of the X chromosome, and knowing this, as you will see shortly, knowing this information was very helpful for us in carrying out this project. So, and here's why. So what we did was, when we did the project, we isolated a number of single cell clones from these from this cell line, and shown here are some sequencing results from 2 of those clones. Okay, so we actually found 3 clones that had all 5 copies of the gene knocked out. Okay, and you can see that here. Some of these are repeated alleles because there were multiple E. coli subclones. But if you were to look closely enough, you would see 1, 2, 3, 4, 5 different alleles of this gene, 5 distinct knockout alleles of this gene compared with the wild type sequence. And the same is true for clone 2. So this, again, was very helpful, having this knowledge that the cell line contained 5 copies of this gene before we did all the subcloning and sequencing, because otherwise we may have had to sequence many more E. coli subclones before we realized, okay, there are probably only 5 copies of this gene. So we strongly recommend, if you're doing cell line work, to incorporate a step using the GeneCopoeia vivid FISH probes to analyze your cell line to determine your gene's copy number in the cells. Okay, and if you want to know more about how useful it is to do a FISH assay on your cell line to determine chromosome copy number, We have an application note on the topic shown here, and I'm going to copy and paste that into your chat box as well. Okay, so I just pasted it into your chat box, so go ahead and look at that when you have a chance. Okay, so moving right along. So let's talk about now transfection or transduction. So again, with cell line work, most people transfect plasmids into cells. Alternatively, people will use lentiviral transduction, especially for hard to transfect cell lines. But in either case, if you're using transfection, these plasmids are very standard plasmids for transfection. There's nothing unique about them. They usually carry standard selectable markers like neomycin for G418 resistance. So we recommend using either GeneCopoeia's end-effectin brand of transfection reagents or whichever works for you. And again, they carry very standard selectable markers and reporter genes. The same is true for lentiviral transduction. This is a schematic of a typical GeneCopoeia lentiviral plasmid. The plasmids are compatible with third-generation packaging cells. And so we recommend using GeneCopoeia's LentiFect packaging reagents or whichever works for you. We can also create the lentiviral particles for you. And again, they carry very standard selectable markers and reporter genes. All right, so the next concept I want to discuss is clonal isolation. Okay, so by clonal isolation, what do I mean by that? Well, I'm talking about isolating single clones, single colonies of cells after transfection, okay, or transduction. And people usually do this by one of two methods. They will plate cells at low enough density to form single colonies and then pick these off a dish, usually with sterile cloning cylinders. And that's the preferred method. It's the cleanest method. But that only works for adherent cells. If you're working with suspension cells, it's a little bit different. We then recommend doing serial dilutions in multi-well plates. Okay, and again, this is very important. I can't emphasize enough how important it is to do clonal isolation because it minimizes the potential effects of unwanted modifications resulting from cell division or off-targeting. So you might be able to analyze a phenotype from a pool of cells that have been treated with CRISPR, but you're going to have a mixed population of cells, and it could just make interpreting your results that much more difficult. So we recommend going through this step of clonal isolation. It takes a long time. It's difficult hands-on work, but it's very important. It gives you much cleaner results. Okay, now I'm going to talk about the screening procedure for edited clones. And I put this very bold statement up front. It's very important to connect your genotype to your phenotype. In other words, we recommend checking the chromosome first before doing functional assays for your target. So, for example, you might be working with a particular gene and you have an antibody to the protein product, but instead of transfecting with your CRISPR plasmids and then checking for antibody to see if doing a Western to see by antibody if your protein has gone away. Let's say you want to knock it out. We instead recommend checking the chromosome to make sure you do have a complete knockout first. And we have had people call us and say, "Hey, I transfected with CRISPR and I did a Western and my protein is still there." Well, the reason could be, there could be several reasons for that. One of which is that you may have only knocked out one copy of the chromosome and you have one or more functional copies left over. So again, connect your genotype to your phenotype, check the chromosome, and I'm going to go over how you can do that. So I'm going back to this slide where CRISPR usually works by creating double-strand breaks. And they're repaired by either non-homologous end joining or homologous recombination. The application that you are using will strongly influence how you go about screening for these modifications. All right, so for non-homologous end joining mediated mutations, this is the general workflow. So we recommend doing your transfection, then isolate clones without selection for 2 to 3 days post-transfection or transduction. Okay. Next, we recommend generating PCR products using primers flanking the double-strand break site, and these can be the same products that were used for validation techniques that I mentioned before. And then you can screen the PCR products using the T7 endonuclease 1 assay or by direct sequencing. And then we recommend rescreening positive clones by direct sequencing. Okay. Now, if you're doing homologous recombination mediated applications, let's say you're knocking in a point mutation or you're tagging a gene, the workflow is quite different. Okay. And so this is how homologous recombination works. You transfect with your CRISPR plasmids and a donor DNA, and the donor copies information from itself into the targeted chromosome. Okay. And so this is a typical GeneCopoeia donor plasmid. It contains usually contains a selection cassette of a marker, of a selectable drug selection marker, and maybe a fluorescent reporter, as well as, and these are flanked by homologous arms. So what we recommend doing to screen for these types of modifications is known as junction PCR. Okay, so but first, again, you mentioned you co-transfect your CRISPR-Cas9 plasmids along with your donor plasmids. 2 to 3 days post-transfection, you apply selection for the donor selection cassette, or alternatively, sort cells that are expressing the fluorescent reporter. Isolate single clones and then screen the single clones for correct integration by this junction PCR method that I just mentioned. So the junction PCR method involves actually making 2 PCR products. Okay. For each PCR product, you have one primer that hybridizes with a region of the DNA that's unique to the donor plasmid. So in this case, it could be like the CMV promoter that's driving expression of a gene of interest, for example. And this is usually not going to be found anywhere in the genome. Okay, but the second primer comes from outside the region of homology, so it can only be found in the chromosome and not in the donor. Okay, so the reason to do this, and you do this at both ends, both junctions of where the donor plasmid integrates into the genome. The reason for doing this is that you will only get amplification by these primers if you had successful integration. Shown here is a typical result. We show double junction PCR from 2 different clones that were isolated for puromycin resistance. Okay, so lanes 1 and 2 come from PCR products from one of these puromycin resistant clones, and we get amplification with both sets of PCR products. So that's good. That tells us that the integration occurred in the correct place. But on the other hand, the 2 PCR products in lanes 3 and 4 are negative. So this tells us that the donor probably integrated randomly because the cells were, again, puromycin resistant. So in order to survive, they had to integrate the DNA somewhere. So they did not integrate it into the correct place. So this is a very important step to take if you're using donor plasmids. We have all the reagents you need. You can use the Indel check kit for that. It contains PCR reagents for that. Okay, now finally, I want to talk about things to look out for, various pitfalls. So the first issue to look out for, to keep in mind, is transfection efficiency. Again, so what do I mean by transfection efficiency? Well, this refers to the percentage of cells that take up the plasmids, and this can influence the modification rate. So now this is a very simplistic thing, but in general, you might say expect to screen twice as many clones for a cell line that has 40% transfection efficiency as you would for one with 80% transfection efficiency. Okay, and so we recommend predetermining your transfection efficiency or using the reporter genes on GeneCopoeia plasmids to get a handle on your transfection efficiency. Now, if your transfection efficiency is really low, then you might want to switch to lentiviral-based transduction. Okay, the second pitfall to look out for, I already mentioned this, but I'll go over it again, is gene copy number. Again, for complete knockouts or mutagenesis, you might need to modify all alleles, but not all cell lines are diploid. And so I don't need to go through these again, but we recommend, again, checking the gene copy number, which will help you tremendously during your screening procedures. The third issue to consider is cleavage efficiency. So I did mention this before. Efficiencies of indel formation by CRISPR varies a lot. It can usually range from 5 to 70%. And so, again, this is a highly simplistic thing, but you might expect to screen twice as many clones for a CRISPR single guide RNA with 40% cutting efficiency as you would for one with 80% cutting efficiency. And so, again, so selection for modifications using a donor can help that screening. So, again, this is all about trying to predict how many clones you might need to screen in your project. We usually recommend screening anywhere from 20 to 100, but these different factors can influence the number of clones you actually screen. All right, so with that, I'm going to summarize. I told you that the workflow for genome editing experiments must be carefully considered and differs dramatically from that of RNAi. Other considerations must be taken into account for successful genome editing experiments, such as transfection efficiency, copy number variation, and cleavage efficiency. And GeneCopoeia offers many products and services, including plasmid design and construction, functional validation, validation and screening kits, and transgenic mice to meet your genome editing needs. All right, so if you want to know more, if you want to get some more detail about how to use CRISPR in mammalian cells, we have a technical note on the topic. This is the download link, and I will copy and paste that link into the chat box. Okay, so I just sent that link along to you. All right, so now before I take questions, I want to tell you about an upcoming webinar. We give CRISPR webinars at least once a month, and the next one will be on Wednesday, March 22nd at noon Eastern Time, and it's called Applications for Cas9 Stable Cell Lines. And this is the registration link, but again, I don't expect you to write this down or memorize it. I will copy and paste that into your chat box as well. And all you need to do is click on that link if you're interested, and then you can register for that webinar the same way you registered for today's webinar. All right, so with that, I'm going to stop and take questions. We have a bunch of good questions. The first is, how do we get a copy of the slides, as we cannot get all the information in one slot, excuse me. Yeah, that's no problem. The slides will be available on our website by tomorrow, as well as a recording if you want to watch the recording again. Next question, are you going to discuss knockout animal models? Unfortunately, no. As I mentioned in the beginning, this webinar is devoted primarily to work using CRISPR in stable cell lines. However, we will have a future webinar on the topic of using CRISPR in animal models. Okay, next question. What length of homology donor sequence do you need to make point mutations? Well, that varies a lot. We usually recommend a minimum of 500 base pairs on either side of the break. That's if you're using a large donor plasmid. You can also use an oligonucleotide. I did not go over that. And in that case, you would want about anywhere from 40 to 80 nucleotides of homology on each arm. Okay, next question. At what point can I verify at the protein level the effect of my gene knockout using CRISPR? Well, again, as I mentioned before, you should check to make sure that you have a complete knockout first by using the indel check kit and sequencing to check to make sure that all alleles of your gene are knocked out in the cell line. Okay, next question. Let's see. Can you isolate clones that joined via NHEJ? Yes, you can. It's not done with any kind of selection, though. You usually have to just you just isolate clones that are without selection, and then you screen those to make sure they have the NHEJ mediated mutations. Okay, let's see. Next question. What is the best way to evaluate any off-target effects? Well, that's a good question. I didn't talk about that, but the best way to check for off-target modification is to do next-generation sequencing on your edited clones. Alternatively, you could use software prediction to predict off-target sites and then check to see if those are modified, but NGS is unbiased, unlike checking predicted sites. So it's really a matter of choice. Let's see. Next question. Is it possible to use this method to knock out pro-survival genes or genes involved in cell proliferation? Yes, it's possible, but if you're knocking out an essential gene, yes, that could be difficult. You might only get single allele modification clones, but you might want to consider using a conditional knockout or some kind of inducible knockout. Let's see. What about in-frame deletions? 1 in 3 times NHEJ should cause in-frame deletions, multiples of 3, and therefore only delete a few amino acids rather than cause a frame shift or nonsense mutation. Is this something that we need to screen for later? Yes, absolutely. That is a very good question. Yeah, as you mentioned, 1/3 of the time when you're doing NHEJ, your indels will be in frame. Now, that could still knock a gene out. You may have knocked out an essential amino acid, but sometimes you may still have a functional protein. So that's another reason why we strongly recommend that you connect your genotype to your phenotype first, and that way you can screen for only clones that have frameshift mutations. Okay, so next question. In functional validation, do you need to run this test on each cell line you will be using to do editing? Well, that's a very good question. I would say you don't have to. It's not essential. You could probably just do the functional validation in one cell line, but we certainly recommend doing it in multiple cell lines if you are in fact working in multiple cell lines, because you may get different results. So yeah, I would recommend doing it in every cell line you're using. How about ssDNA as an SHR donor? How efficient and how to select? That's a good question. I didn't go over single-strand DNA as a homologous recombination donor, but you would screen that the same way you would for NHEJ mutations, basically doing sequencing to look to make sure you had your modification. Next question. If we select clones, shouldn't any progeny have the same mutation and in essence be a stable cell line? The answer is yes. If you've got clean isolation of a cell clone, then yes, each one, all the progeny cells will have the same mutation. Although you can get recombination, you can get gene conversion if you only had single allele modification that would repair it to wild type. But in general, the answer is yes. Okay, next question. If I want to do a point mutagenesis to, say, constitutively activate a transcription factor, is there an advantage to using CRISPR over a standard method? And what is the workflow? Does the guide RNA incorporate the mutation? No, the guide RNA does not incorporate the mutation. For that, you would use a donor, just like with any other homologous recombination application. And then you would just screen it in the normal way using an oligonucleotide. Okay, so next, with regard to the homology arms for the donor plasmid, how long do you suggest each arm to be? Well, like I said, as I mentioned before, minimum of 500 base pairs on each arm if you're using a plasmid, although longer is better. Okay, next question. What is the benefit of using a donor clone for mutation instead of simply knocking out the gene using CRISPR and then making a stable cell line with another vector that contains the mutated gene sequence? Well, that's a good question. You can get knockouts using just NHEJ. It might sometimes be advantageous to use a donor plasmid for getting knockouts. And the reason being is that NHEJ mutations are unpredictable, so they're kind of random. But if you wanted to make a very specific deletion, for example, then you would want to design that with a donor clone. And also, donor clones give you the benefit of selection, which NHEJ does not. Okay, next question. Do you have any tips for designing sgRNA for knockout using long non-coding region? Yeah, that's a good question. There's been a lot of interest lately in knocking out long non-coding RNA and coding genes. Usually, we recommend using 2 guide RNAs that will delete all or most of the LNCRNA sequence. But that's something we can talk about if you're interested and if you want to contact me. Next question, is sorting really important? Yes, sorting can be very important, especially if it's the only way you can do single clone isolation. So again, it's usually part of the single clone isolation workflow. It can also enrich your cells, enrich your population for transfected cells. Okay, in terms of CRISPR, if we knock out a gene, then should we go for HR or NHEJ? Again, I already kind of answered that question. It depends if NHEJ occurs at higher frequency than HR, but HR gives you the benefit of selection usually. Okay, so with transfection, Cas9 is constitutively expressed. Would the Cas9 keep editing genes throughout the cell culture? The answer is potentially yes, but if you're doing NHEJ, then it will and you designed the CRISPRs properly, then once it has modified your gene, then it can no longer modify your gene because the target sequence has been mutated. If you're using a homologous donor clone, then it's usually best to design the donor clone so that it cannot be cut by CRISPR. And so if everything is properly designed, then the answer is no, it will not keep editing. Okay, next question. What ratio of sgRNA to Cas9 donor plasmid should be used for transfection? That's a question we do occasionally get. We usually recommend starting out with a 1-to-1 molar ratio and then optimizing from there if necessary. Okay, next question. In the presence of a donor clone, what is the probability of having homologous recombination versus NHEJ? Well, that's highly variable. NHEJ can range from, say, like I mentioned before, 5 to 70%. Homologous recombination frequencies are usually lower. They're usually maybe 3% to 5%, but it varies a lot. Okay, next question. Do you recommend using ganciclovir to get rid of random integration? That's a good question. If you're really concerned about random integration, then yes, you might want to use ganciclovir. Some of our donor clones have the thymidine kinase gene on it, but it's usually not necessary. If everything is working well, then CRISPR causes a very large increase in the rate of stimulation in the rate of homologous recombination. Okay, next question. Is it possible to make sure Cas9 cuts the target sequence only once and that the plasmid is not integrated in the genome, thus increasing the rate for off-target effects? Yeah, like I said, it's very important to do the screening methods that I mentioned before. Cas9 will not cut the chromosome anymore once it has made the cut if you get NHEJ. And if you are using a donor clone, well then by all means, please do the double junction PCR method that I mentioned earlier for screening for correctly integrated clones. Okay, next question. When using the plasmid pCRISPR-CG01, that's one of the plasmids I showed before, you'll obtain a transient transfection, or is it possible to select also stable transfected clones? Yes, with that plasmid has the neo gene on it, so you could select for stable clones if you wanted to. We usually recommend not doing that, but it might be useful to do that in some cases where your transfection efficiency is low. Okay, next question. If gRNA cuts DNA in an intron, will it knock out the gene or leave it completely functional? Well, that depends. If you, yeah, sometimes the having the guide RNA cut in an intron will leave the gene functional if it only repairs the intron. Now, if you have it close to the intron exon junction, then you might disrupt that junction and disrupt splicing, and so it will knock out the gene that way. And sometimes you even get large deletions that can be a couple hundred base pairs. So really the answer is it depends on what's going on. Let's see. Next question is, does GeneCopoeia supply the primer sequence for functional validation? We can do that if you want. It costs a little bit of extra, but we can work with you on that. Next question, how do I determine if both the alleles are knocked out if my gene copy number is 2? Well, again, doing clonal isolation and sequencing on your edited clones will tell you that. And you need to do, when you do the sequencing, you need to subclone the PCR product into E. coli so that you identify sequence multiple E. coli subclones. And that will tell you if you have both alleles knocked out. Because what you'll usually see by NHEJ is if your gene copy number is 2 and you get both alleles knocked out, then you will see 2 different indel mutations. Because again, NHEJ is random. So you rarely, if ever, get the same indel mutation on both chromosomes. Okay, next question. When sorting single cells by flow cytometry, do you recommend some staining to differentiate live or dead cells? No, that's probably not necessary because you're going to grow these out anyway. I mean, if you saw a lot of cell death, maybe, but I don't think that's really necessary. I think you should just sort the cells and then try to culture them, and then maybe in 96 well plates, and then if there's anything alive will grow up and anything dead will not. So, but it's a good question. Okay. Okay. Are there any more questions? Got lots of good questions in this webinar. Anybody? All right. Well, if there are no more questions, then I'm going to stop. And if you have any more questions, feel free to give me a call at 866-360-9531, extension 227. Or feel free to drop me an email, edavis@genecopoeia.com. And please visit our website, www.genecopoeia.com, and check out all the wonderful genome editing products and services we have. Okay, so with that, I'd like to wrap it up and to thank you for joining us for today's webinar and to have a great day.